Hello everyone, this side Dr.G and today I will be discussing about the second week of development, that is the formation of bilaminar germ disc. So let's start. We have already discussed in the first week about the blastocyst, right? In the first week we know we have the outcome was the blastocyst, right? So in the second week, what are the events that will occur? So first I will tell you what are the events which will occur during the second week, and then I will draw and explain to you. So remember, the following changes will occur during the second week. It will occur during the second week. Now, as you already know, it is the second week of development, right? Second week of development. So what will happen? You already know the implantation has started on day 6. But remember, the implantation will be completed by day 10. And day 10, it is the second week of development, right? Day 10 to second week mein aa gaya, right? So day 10 will be the second week of development. So the first thing that will happen, that is the implantation. Implantation will complete, right? Remember, it is completing by the 10th day, right? So implantation means the blastocyst completes its implantation. Blastocyst completes its implantation by day 10, right? You already know it starts at day 6. So initially in the first week it was started, and by day 10 it will get completed, and day 10 it is in the second week. This is the first change that will occur, right? Apart from that, you already know we have something which is called as the trophoblast. Okay, now you already know the structure of the blastocyst. Let me draw for you. You know we have the outer cell mass, right? We have the outer cell mass which is arranged in the periphery, right? And along with that, at one corner of the cell we have the inner cell mass, right? We have this inner cell mass. Because of the rupture of the zona pellucida, what will happen? The uterine fluid will be this structure will absorb all the uterine fluid, so it will push the inner cell mass to one of the sides, right? What will be created? A cavity will be created, and that cavity it is called as the blastocele, right? And this structure it is called as the blastocyst. So you already know this is the blastocyst, and you already know this is the outer cell mass, this is the inner cell mass, and this is the blastocele, right? This is the blastocele, the one with a cavity, right? So it is called as the blastocele, and the entire structure it is called as the blastocyst. Now what will happen? This outer cell mass in the second week, right? In the second week it will be called as the trophoblast, right? It will be called as the trophoblast in the second week of development, right? And this trophoblast will differentiate into, okay, it will differentiate into, remember, Syncytiotrophoblast and Cytotrophoblast. We have Syncytiotrophoblast and Cytotrophoblast. I will be coming to the structures of both these, so no need to worry. I'm just giving you an overview right now. What changes will be there? Cytotrophoblast. So in the first week we have the outer cell mass. Now this outer cell mass will be called as the trophoblast, and this trophoblast will differentiate into syncytio and cytotrophoblast. Apart from that, this inner cell mass, okay, this inner cell mass in the second week will be now called as the embryoblast, okay, it will be called as the embryoblast, right, and it will also differentiate into two layers, okay, it will also differentiate into two layers, and remember that these two layers are the epiblast and the hypoblast. Epiblast and the hypoblast. Now remember, the epiblast, yes, the epiblast, it is called as the primitive ectoderm. Primitive ectoderm. It will form the ectoderm in the future. See, in the second week, no ectoderm is there. It is the primitive ectoderm which is called as the epiblast. Apart from that, the hypoblast will be called as the primitive endoderm. It will be called as the primitive endoderm, right? So that's why always remember when we say second week, so it is an easier trick to remember. Second means two. So two things are happening everywhere. The outer cell mass, that will be the trophoblast, it will be differentiating into two layers, syncytio and cytotrophoblast. The embryoblast, that is the inner cell mass, it will differentiate into epiblast and hypoblast, again it is two. And apart from that, remember one more thing, that two cavities will be formed, okay, two cavities will be formed. Now, which two cavities will be formed? These are the amniotic cavity and the yolk sac. So remember, these are the amniotic cavity and yolk sac. So everything like it is two in number, so it is easier to remember, right? So when I say bilaminar germ disc, so I'm actually talking about this epiblast and the hypoblast, right? Bilaminar germ disc, it simply means the epiblast and the hypoblast. Along with this change, there are other changes also which I have discussed on the screen. Now let us see how this process goes on. Now, what will happen? The first thing which is happening over here, it is that now you already know the structure. Let me just draw for you people so that it is in continuation. You know that this is the outer cell mass, right? This is the outer cell mass. And we know inside we have the inner cell mass. Right? So remember, the first thing happening in the second week, it is the cells of the inner cell mass. Now this is the inner cell mass, you already know, inner cell mass. The cells of the inner cell mass which are facing towards this cavity. Now cavity का नाम क्या है? Which are facing towards this cavity, which is like the blastocyst, right? You already know this cavity over here. इस cavity का नाम क्या है? It is the blastocyst. So the inner cell mass, right? The cells which are facing towards the cavity, the one which I am highlighting right now. These cells, okay, ये भी inner mass cells ही हैं. So the one which are facing towards the blastocyst or towards the cavity, they will give rise to the first primitive germ layer, okay, the first primitive germ layer, and that is called as the hypoblast, right? That is called as the hypoblast. So let me just draw it. So outer cell mass I will be now drawing as a single line, but remember beta, it is the cell only, right? And it is inconvenient to draw every time the cells. So yes, let me just draw it. So this is, like you can see over here, this is the outer cell mass. Right? So this outer cell mass after implantation will be now called as the trophoblast. Okay, it will be now called as the trophoblast, right, after implantation. After implantation, right. Now these cells have changed and this will give rise to the hypoblast, right. Let me draw it with green color so that it's easier to notice that yes, we have these hypoblast which will be formed, right. So this inner cell mass will be changed into these cells, and these are called as the hypoblast, the cells which are facing towards the cavity, right, and yes, that cells will be now called as the hypoblast. Now the remaining inner cells, now when I say the remaining inner cells, I am actually talking about the other cells which are remaining, the one which I am highlighting with green. So these are the remaining inner cells mass. Now they will all presume the tall columnar shape, right, they will all presume the tall columnar shape. So yes, and they will be now called as the epiblast, right. So yes, they will all presume this tall columnar shape, and this is now called as the epiblast. Epiblast. Now you already know the epiblast it is the primitive ectoderm. It will form the ectoderm later on. So it is the primitive ectoderm, and the hypoblast, it is the primitive endoderm. Primitive endoderm, right. So that is the first change which is happening, right. Now, so if I ask you, there are two questions that you have to remember over here. The first primitive germ layer to be formed, right. If I ask you the first primitive germ layer to be formed, and that is called as the hypoblast. That is called as the hypoblast, right. The second one, the second primitive germ layer to be formed, it is the epiblast. Epiblast or primitive ectoderm. Now if I ask you, hypoblast and epiblast, they both are coming from inner cell mass. It is very important to trace the pathway कि वो आ कहाँ से रहा है, ठीक है. तो you already know they both are coming from the inner cell mass, right. Now what will happen now? What change will occur now? So this is the first structure that we have drawn. Now let us try to see what next will happen, right. Now let us draw. Now what will happen? These epiblast cells, now the epiblast cells are these tall columnar cells, right. Now these epiblast cells will give rise to embriogenic cells, right. These epiblast cells will give rise to embriogenic cells. So we have this particular diagram. So let us draw this diagram again and understand. Now let us suppose this is the outer cell mass, right, or it is now called as, better to write it as trophoblast. Now this is trophoblast. So I'm just showing you the inner change development right now, inner cell mass differentiation right now. So we already know we have the hypoblast over here, right. Hypoblast, please follow the color code, whichever color like I'm drawing, the same it will be continued later on also. So the hypoblast I have drawn it with yes, the green color, right. So remember this is the hypoblast. So remember this is the hypoblast, and on the top you already know we have this epiblast over here, the tall columnar cells, these epiblast. Now these epiblastic cells or these epiblast will give rise to another cells which will line the trophoblast from inside, and this cell, right, which you can see, these are called as the embriogenic cells. These are called as the embriogenic cells, and remember these embriogenic cells, they are derived from epiblast, right? They are derived from epiblast. So remember the epiblast will give rise to a cellular layer, which is cellular layer above, which is lining from inside, right? And again there will be a cavity which will be formed, and remember this cavity, it is called as the amniotic cavity, right? This cavity, it is called as the amniotic cavity. Amniotic cavity. Now coming on to the next one, that is the hypoblast. Now these hypoblast cells will also give rise to a cellular layer, right? Now they will also give rise to a cellular layer which will cover the blastocele from inside, right? It will cover the blastocele from inside. You can see hypoblast के नीचे क्या था, blastocele था, right? And this hypoblast, it is also giving rise to a cellular layer which is covering the blastocele from inside, and remember this is called as the Husser's membrane. Husser's membrane. Husser's membrane, right? And if I ask you the Husser's membrane, it is derived from hypoblastic cells or the hypoblast or the hypoblast. Again there will be a cavity which will be created in between, and this cavity remember it is termed as the yolk sac. It is termed as the yolk sac. So over here I have completed the basic structure. See beta, two cavities are formed. I have already told you in the starting कि the two cavities which are formed over here, you are very well aware, this is the yolk sac, right, and the amniotic cavity, Apart from that, I have already discussed about the epiblast, right, and the hypoblast. Now if I ask you epiblast and hypoblast, they are arising from where? They are arising from the inner cell mass, right? Inner cell mass will be now called as the embryoblast after implantation, right? And they are giving rise to epiblast and hypoblast. Now if I ask you the embriogenic cells, they are coming from epiblast. The Husser's membrane, it is coming from hypoblast. So this is like you have to remember all this in sequence, right? Now coming on to the next part, So I hope till now everything is clear. So let us just discuss what will happen next. Now what will happen next? Now the yolk sac, right? The yolk sac or the wall of the yolk sac, basically what will happen, this yolk sac will secrete a lot of secretions, right? It will secrete a lot of secretion, right? So the walls of the yolk sac, or I can say this is the primary yolk sac. It is the primary yolk sac. The walls of this primary yolk sac will proliferate, okay, and the cells they will accumulate outside. So what will happen? Let us just draw this. Now let us suppose this is the trophoblast. This is the trophoblast, right? Now To make it simple and easier, what I have done, this whole blue color, yes, this whole blue color I will be like drawing in a single line like this, right? I will be drawing in a single line. Now this is nothing but the embriogenic cells and the epiblastic cells. Apart from that, I will be drawing this, the hypoblast and the Husser's membrane in a single line like this. So just to make it more clear, you already know over here, this is the amniotic cavity, right? Amniotic cavity, and this is the yolk sac. This is the yolk sac. Now what will happen? The walls of the primary yolk sac, or this is the primary yolk sac, will proliferate and the cells will like accumulate outside it. Now they will start proliferating. Again, this is an update that you have to remember. These are the walls which will proliferate and will accumulate outside. Now what will happen? They will give rise to small pouches outside, right? These small cavities will be formed. These small cavities will be formed all around, right? Right. So yes, and this wall of the yolk sac, these cells which are actually the cells which are proliferating outside, they will give rise to one of the mesoderm which is outside, and this mesoderm which is outside, and in between, because the cells of the yolk sac, the cells which are coming from the wall of the yolk sac, they are dividing, right? They are dividing and they are giving rise to this particular what we say, extraembryonic mesoderm, right? So they are giving rise to this extraembryonic mesoderm, the one which I am highlighting in the yellow color, right? Now this yellow color, right, it is the extraembryonic mesoderm. Extraembryonic mesoderm. So remember this extraembryonic mesoderm. देखो एम्ब्रियो तो अंदर से बना है, एम्ब्रियो ब्लास्ट तो ये है ना पूरा का पूरा ब्लू और ग्रीन, एम्ब्रियो ब्लास्ट जिसको हम लोग बोलते हैं, जिसमें हमने एपीब्लास्ट, हाइपोब्लास्ट, ह्यूसर्स मेम्ब्रेन और एम्ब्रियोजेनिक सेल्स पढ़ी, ठीक है। इनर सेल मास इज द एम्ब्रियोब्लास्ट। उसके बाहर वाला जो भी होगा, आउटसाइड इट वट एवर विल बी देयर, इट विल बी द एक्स्ट्रा एम्ब्रियोनिक मेसोडर्म ओनली, राइट। अगर इस एम्ब्रियोब्लास्ट के अंदर मेसोडर्म बनेगा तो मैं क्या बोलूंगा, इंट्रा एम्ब्रियोनिक मेसोडर्म। बट the mesoderm which is outside and it is coming from the walls of the yolk sac, it will be called as the extra embryonic mesoderm, right? So this extra embryonic mesoderm, again this is an update, it will come, or it is derived from wall of the yolk sac. It is derived from wall of the yolk sac, right? This is again an important question. Apart from that, in this extra embryonic mesoderm, what will happen? These small cavities will be formed, right? These small cavities will be formed, right? And later on, these all cavities will be like joining together. Now, how these cavities will be formed? देखो, mesoderm है, cells हैं, the cells will secrete something and there will be small cavities will be formed in this extraembryonic mesoderm, and later on these small cavities, they will unite with each other, they will join with each other to form a single cavity outside, and outside this embryoblast, that is called as the extraembryonic coelom. coelom का मतलब ही cavity होता है। एक्स्ट्रा एम्ब्रियोनिक मेसोडर्म के अंदर ये कैविटी बनेगी, दैट इज कॉल्ड एज द एक्स्ट्रा एम्ब्रियोनिक सीलोम। सो लेट अस जस्ट ड्रॉ इट एंड ट्राई टू लर्न ऑल दिस, राइट। नाउ व्हाट विल हैपन, लेट अस सपोज, नाउ लेट अस ड्रॉ अ बिग डायग्राम सो दैट यू ऑल अंडरस्टैंड दिस, राइट। सो रिमेंबर दैट दिस इज द ट्रोफोब्लास्ट, राइट, दिस इज द ट्रोफोब्लास्ट, right? Trophoblast कहाँ से आया? It is the outer cell mass, right? After implantation, outer cell mass is called as the trophoblast. Now, we have the primary yolk sac, right, and the amniotic cavity. So yes, the amniotic cavity along with the embriogenic cells and the epiblast cells, I am representing this by this. This is the amniotic cavity. This is the amniotic cavity. We have, yes, yeah, I have drawn it with green color. So please be very clear with the color coding so that it's like easier for you to understand later on. So this is called as the yolk sac. This is the yolk sac. Now the wall of the yolk sac, इसके wall से क्या निकल रहा था? The cells will proliferate and will give rise to a mesoderm, which is outside this embryoblast, right? And that's why it is called as the extraembryonic mesoderm, right? That's why it is called as the extraembryonic mesoderm. Now, what will happen? A cavity will be formed. Remember, a cavities will be starting development in the extraembryonic mesoderm, and these cavities, remember, these cavities will join together to form a large cavity, and that is something which is called as the extraembryonic coelom. It is called as the extraembryonic coelom. Now, you already know this extraembryonic coelom, it is developing where? It is developing in the mesoderm, right? It is developing in the mesoderm. And which mesoderm? वो embryo का mesoderm है? नहीं, वो embryo के बाहर वाला mesoderm है. So that's why it is called as the extraembryonic mesoderm. So let me just highlight this extraembryonic mesoderm. So yes, please use pencil colors for drawing all these diagrams which I am drawing, okay, which I am drawing with you people. So yes, highlight this. Yes. Now you will be surprised to know कि extraembryonic coelom I have drawn all around, but I have left one side open. One side, like on the top, I am not drawing it. Why? What's the reason? The simplest reason is, beta, you already know there is some undifferentiated extraembryonic mesoderm. No, there is something which is connecting this embryo, right? So that connection, it is called as the connecting stalk. वो connecting stalk यहाँ पे present होगा, इसलिए वो सीलोम वहाँ पर absent है, ठीक है? वो सीलोम वहाँ से absent है, which is actually holding the embryo. और वही connecting stalk, it is later on forming what? The umbilical cord. It is later on forming what? The umbilical cord, right? So yeah, just highlight it completely so that yeah. Yes. So yeah, just highlighting it so that it's clear to you, right? Now, this yellow color which I have highlighted, it is the extraembryonic mesoderm, right? I'll just mark it so that it is clear to you people. It is the extraembryonic mesoderm, right? Now, extraembryonic mesoderm, it is coming from the walls of the yolk sac. It is coming from the walls of the yolk sac, right? Now, what will happen? I have told you this extraembryonic coelom will be absent over here. So there is something which is called as the undifferentiated mesoderm. Undifferentiated mesoderm, right? This mesoderm, it is not having this extraembryonic coelom, right? And this is something which is later, like it is the connecting stalk, which will later form the umbilical cord. Okay, it will later form, later on it will form the umbilical cord. Umbilical cord. Now this extraembryonic mesoderm will be now divided into two parts, somatopleuric and splanchnopluric. Now, somato means Somato means basically the body wall, right? It is called as the body wall. So everything which is towards the body wall, can you see this outer cell mass, right? The trophoblast. I'm saying it as outer cell mass, but remember it is the trophoblast. Yes, it is the trophoblast. Now, towards the mesoderm, which is towards the body wall, it will be called as the somatopleuric. सोमैटो का मतलब ही क्या होता है? बॉडी वॉल, राइट? सोमैटोप्ल्यूरिक एक्स्ट्रा एम्ब्रियोनिक मेसोडर्म, एक्स्ट्रा एम्ब्रियोनिक mesoderm, right? Somatopleuric extra embryonic mesoderm. And the one which is towards the embryoblast or towards the yolk sac specifically, देखो mesoderm जो अंदर की तरफ है, one which is outside this extra embryonic coelom, it is the somatopleuric which is all over present towards the body wall. But the one which is towards the yolk sac, specifically beta I am using the term yolk sac बिकॉज योक सैक ही फ्यूचर में जाकर क्या बनाएगा? इट विल फॉर्म द फ्यूचर गट, द फोर कट, मिड कट एंड हाइंड कट. You already know the foregut, midgut and hindgut, they will lead to the development of the visceral organs, right? They will lead to the development of the organs of the body. So it is very much clear over here कि the mesoderm, अंदर वाले सारे mesoderm को हम लोग splanchnopleuric नहीं बोल सकते. The area has to be specified, and this area which I am highlighting. It is just towards this yolk sac, right? And this is called as the splanchnopleuric mesoderm. Splanchno का मतलब ही क्या होता है? Visceral. Okay, it means visceral. It is towards the body viscera, right? Towards the visceral organ. So remember, this is the splanchnopleuric extraembryonic mesoderm. extraembryonic mesoderm. Obviously बात है, एम्ब्रियो के बाहर है, right? But it is up till the yolk sac, right? It is up till the yolk sac, right? Now, remember two important things over here. Now, I'm going to tell you about the two important structures. One is the amnion and one is the chorion, right? So the one which I am marking right now, this is called as the amnion. This is called as the amnion. Now, अब ये amnion है क्या? You already know it is the amniogenic cells. I have told you कि amniotic cavity को जो line कर रहा है बाहर से, it is the amniogenic cells. So amnion, it is the amniogenic cells plus you can see which mesoderm is there, it is somatopleuric extraembryonic mesoderm. It is somatopleuric extraembryonic mesoderm. EEM I am writing. Somatopleuric extraembryonic mesoderm. Now, chorion is nothing but this structure. Yes, this structure. So this is something which is called as the chorion. And remember, the chorion, it is nothing but the extraembryonic mesoderm. And which extraembryonic mesoderm? It is the somatopleuric extraembryonic mesoderm plus this black one, that is the trophoblast, right? So we have the extraembryonic mesoderm, and which one? It is the somatopleuric towards the body wall. In both the cases, somatopleuric is there. चाहे amnion हो, चाहे chorion हो, it is the somatopleuric extraembryonic mesoderm plus the trophoblast. So if I simply ask you what is chorion, it is somatopleuric extraembryonic mesoderm plus the trophoblast. And if I ask you what is amnion, it is somatopleuric extraembryonic mesoderm plus the amniogenic cells, right? So that is in nutshell about this. Apart from that, you have to remember one more thing, one more important thing, Now, we already know that there was earlier primary yolk sac, okay? There was earlier primary yolk sac. Now, what will happen? What will happen? The yolk sac, okay? The yolk sac which is inside, right? The primary yolk sac. Now, outside you already know there is the extraembryonic coelom. Now, this extraembryonic coelom will grow larger in size, okay? It will grow larger in size. Now, it will try to compress this primary yolk sac. Now, as the primary yolk sac is compressed, it will take a small portion of the primary yolk sac out, which will be like dissolved later on, and the left yolk sac will be now called as the secondary yolk sac. It will be now called as the secondary yolk sac, right? It will be now called as the secondary yolk sac. Now, so whatever yolk sac is remaining, right, after like the one which has been removed, that is called as the secondary yolk sac. So in this I have discussed about, yes, the embryoblast, the epiblast and the hypoblast, right? I have discussed about the cavities, that is the amniotic cavity and the yolk sac, right? Now coming on to the trophoblast. Now, I'm just focusing on the trophoblast as of now. You already know. Let us just try to draw the diagram in a nutshell. Now, this is the trophoblast. This is the trophoblast, and you already know inside we have the amniotic cavity and the yolk sac, right? So let us just try to draw this. You already know. I will not mark it again. Now, yes. Now what will happen? This trophoblast will now divide into cytotrophoblast and syncytiotrophoblast. Cytotrophoblast and syncytiotrophoblast. Now, trophoblast, it is nothing but the outer cell mass only. So let me just change the ink. Yeah. Trophoblast, it is the outer cell mass only, right? Now, after implantation, it is called as the trophoblast. Now, this trophoblast will divide into cytotrophoblast and syncytiotrophoblast. It will divide into two cells, that is cytotrophoblast and syncytiotrophoblast. Now, what is the meaning of this? Why there is a need of this? Why there is a need of this division? You have to understand. Now, you have to understand outside what is present. Outside what is present, you already know. Outside what is present, that is the uterine endometrium. Yes, outside what is present, this is the uterine endometrium in which this implantation takes place. And when the implantation takes place, the uterine endometrium will be now called as the decidua. It will be now called as the decidua. So you can just highlight this. So yes, remember it is the decidua which is present over here. Yes, this is the decidua. Decidua is nothing but the uterus का जो endometrium है, तो उसी को हम लोग implantation के बाद we give it a special name that is called as the uterine endometrium, that is the decidua. Uterine endometrium after implantation. After implantation, right? Now, what will happen? There will be cytotrophoblast inner, and the cells, some cells of these trophoblast will lose their plasma membrane. Okay, some cells of these trophoblast will lose their plasma membrane and only nuclei will be there. Okay, and why there is a need of syncytio का मतलब क्या होता है? That is only nucleus. Only nuclei are present. There is no plasma membrane. That is for a reason. Why? Because they can help to invade inside the uterine endometrium. अगर सेल की लेयर इंटैक्ट है तो यूट्राइन एंडोमेट्रियम में इन्वेड नहीं कर पाएगा. फॉर द अर्ली न्यूट्रिशन इट इज़ वेरी वेरी इंपॉर्टेंट कि सम cytotrophoblast, these cells, some trophoblastic cells, right? This is the trophoblastic cells. Now, some of these cells, it will lose their plasma membrane. It will lose their plasma membrane and they will come out like this, right? They are losing their plasma membrane and they only have nucleus in it. So, it is the cytotrophoblast which is having their cell membrane intact. The one which has lost its cell membrane, it is called as the Syncytiotrophoblast. Syncytiotrophoblast, right? So now that is the reason, so that they can grab all the nutrition that is present in the uterine endometrium. You already know there are blood filled lacunae, okay? There are uterine vessels, right? So if I just want to draw, you already know we have the uterine arteries, right? So these uterines are actually, there are lacunae which are present all over in the endometrium. So yes, if I just want to draw it, there are lacunae which are present over here, right? There are these lacunae. Now these lacunae, okay, they are glycogen filled, and like these lacunae are required for first 1.5 week. They are required for first 1.5 week. So yes, if they just like yeah, so these cells can absorb the nutrition from these lacunae, and these lacunae will receive all the things from the uterine blood vessels, right? So yes, that is the need over here for this division of trophoblast, so that the nutrition to the early embryoblast can be provided with the help of this, right? Thank you.